Hello. All right, everyone. Well, welcome to today's Monday seminar. It's my great pleasure to introduce my friend, Matt Thomson from Caltech, who I first heard of from my old advisor, Wendell, who described him to me as someone you want to talk to about your data, no matter what kind of data it is. And I think that's really because Matt is a very deep thinker about sort of foundational ideas in biology and biological sciences, which means he always has thoughtful things to say about whatever type of data you're working on. And Matt comes from doing both his undergrad and PhD at Harvard, where he used a blend of theory, data analysis, and experiment to understand biological systems decision-making from a sort of dynamic systems point of view. And this would range from questions about, say, multi-stability in phosphorylation systems to how pluripotency factors control stem cell identity. And then I came to know him when he was a theory fellow at UCSF, where he was exploring tissue organization through the lens of both theory and experiment. And he started his own group at Caltech in 2018, interested in sort of foundational principles govern biological self-organization across scales, from force generation at the level of motors, like we'll be hearing about today, all the way to embryonic development. So without further ado, take it.
Awesome, thanks, Scott. So it's really great to be here. It was so fun to meet with the students this morning. It's been great. It turned out there's this strong Wisconsin-UCSF connection, so it's been great to see everybody. Like Scott said, so my group at Caltech, we do a bunch of different stuff. We're really a computational biology group, and our main focus is collective behavior across scales of organizations. We're interested in how molecules build machines, how cells build tissues. But today I want to talk about kind of work that I'm very passionate about, which is really understanding how the cell generates and controls physical behaviors, which I think are kind of the essential for these things that we think about as the mechanical programs of life. And so one of the things I'm thinking about is the students in the audience, because when I was at your guys' stage, I came from physics into biology, and I spent a ton of time thinking about how cells process information. And there's this kind of paradigm of the cell as being a kind of molecular computer that takes information from the outside, uses gene circuits to process the information and make decisions. But then you see a movie like this. This is a famous movie by David Rogers of a neutrophil, an immune cell, chasing a bacteria. And what strikes me about this movie, there's the bacteria, is that the cell is really like much more than a computer. So it's really this, what I think of as an active computer. It's this machine that can take in information, but really importantly, generate and control the forces inside it to act in the world, here to chase. And so one thing that's been really striking for me, coming from physics, thinking about information processing, is that in many ways, I think the physical properties of the cell, its ability to kind of sculpt geometry, sculpt force, control force, achieve action on micron length scales is even more impressive than the cell as a computer. And so the paradigm that we've been thinking a lot about in the lab is this idea that the cell and a lot of the kind of really fundamental programs of life are active programs. They involve information coming into a cell in the form of a chemical gradient, and then the cell achieving actuation through micron scale self-organization of protein machines. Here, the leading edge of this neutrophil. And this principle of sort of active computation crosses what we might consider fundamental cell behavior. So I just showed you motility. The same thing is true really with self-replication. So this is a movie of a sea urchin embryo. The chromosomes, the DNA are in yellow, the microtubules are in blue. And again, what you see in this movie is the ability of a cell to take in information from the outside world. It's a good time to divide. And then the cell self-organizes this machine, the mitotic spindle. So it self-organizes this force-generating machine that ultimately consumes energy and pulls the chromosomes apart, enabling self-replication. And so again, we have this paradigm. Cell is active computer, information, actuation, and I'll give you one more example. This is a movie of a Drosophila embryo undergoing morphogenesis. And the first time I saw this movie, I was a grad student, and I learned a lot about patterning and morphogen gradients and bicoid. And the thing that struck me is I was like, what's going on? Why are all the cells moving around? The most striking aspect of this movie is really the physics. The cell is able to sculpt its internal shape, generate forces, ultimately egress and bend to undergo gastrulation. And so again, we have cells they're receiving information in the form of chemical gradients, and then they're acting, they're generating internal forces to move and migrate. And thinking about synthetic biology, I would argue that synthetic biology has really made unbelievable progress on the information processing front of things. So we can build remarkable systems like the SynNotch system that really allow us to manipulate how cells receive and process information. But I would argue we've made much less progress so far on actuation. And so while we can control information processing, I would say that our ability to control action and control sort of micron scale generation of force and geometry is much more primitive. And I'm interested in this because I've been thinking a lot about like the technology that can come out of biology now. And I would argue that synthetic biology has made amazing strides in using information processing and sensing to cure disease. But if you look at a lot of human technology, here's a very like a 20th century human technology, an airplane. Human technology is not able to replicate any of these fundamental programs of life. Like we don't have machines that can self-replicate. We don't have machines that can control their molecular composition to control their shape. We don't have machines that can really generate autonomous motion at micron length scales. And so the fundamental goal of this part of my research group is really to understand this interplay between mechanical programs and information processing. And our kind of orienting holy grail is to be able to build synthetic minimal systems outside cells that can reconstitute sort of what we view as fundamental sort of living programs. And so how do we start? So one thing that I think is really remarkable and amazing about these mechanical programs inside cells is that they're all achieved through a kind of universal hardware. And so there are these motor filament systems. There are microtubules and kinesin motors, actin, and myosin motors. And if you look across all the behaviors I showed you, sort of shape change, replication, motility, in eukaryotic cells, the amazing thing is that these systems underlie all of them. And so the systems I want to talk about today are these motor filament systems. Here's a canonical example. This is an illustration of a kinesin motor protein, which has been dimerized. There are 2 copies of kinesin motor protein that are linked. And then here's a filament, a microtubule filament, which is a polymer of a bunch of individual tubulin units. And the way these cellular machines work, these sort of micron scale force generating machines, is through sort of a mechanism that's very different than sort of how we build machines. These motor proteins, as one example, consume ATP on molecular scales. So these motor proteins are like nanometer size. They consume ATP, and that allows them to step along these filaments. And so they convert the chemical energy into force. And the same thing is actually also true about these filament proteins. They're constantly shrinking and growing as they bind GTP and hydrolyze it. And then these nanometer-sized components undergo self-organization to generate a structure called the mitotic spindle, which can actually span many microns in size and length scale. And so you have energy consumption at molecular scales flowing up through self-organization to micron length scales, and then the cell uses this machine to generate the forces that allow it to do replication. And one interesting thing about these cellular machines is that they work very different than the kinds of things we build in human engineering, because energy is being consumed at molecular scales and flowing up to macroscopic length scales through self-organization. When we have something like a turbine, what we typically do is we burn fuel at macroscopic length scales. That fuel turns a macroscopic object, a turbine, and then energy actually flows out into the world, drives fluid flows, and energy is dissipated at microscopic length scales. And so energy flow is going from micro to macro and macro to micro. And so the principles of these non-equilibrium cellular machines are fundamentally different from the principles that we are interested in, that we typically study in human engineering. Another interesting property of these systems is that, so as I said, the motor filament systems, I just want to emphasize, they underlie many, many of the mechanical behaviors of life. I'm showing you 3 here. So microtubules and motor proteins self-organize the spindle, which is the thing that achieves division of the chromosomes during cell division. These cytoskeletal networks are also involved in establishing and modulating the shape of a cell, here in axon growth cone. And then motility, one mechanism of motility in cells is through the polymerization of actin networks at the front of the cell. And so these actin networks also consume energy, polymerize to drive the cell forward. And so a fascinating thing about eukaryotic cells, and actually prokaryotic cells as well, is that these motor filament systems really, I would argue, represent a fundamental hardware that achieves many, many different sort of physical mechanical behaviors that are required for the cells to do sort of all of the magical living programs of life. And another thing to just think about is these are all cell scale, these are cell scale behaviors, but ultimately if you think about an organism like the blue whale, which is 100 meters in length, its swimming is driven by the contraction of muscles, and those contractions are ultimately powered by myosin motors walking on actin. And so the swimming of a blue whale is ultimately powered by ATP hydrolysis and motor proteins that are 10 orders of magnitude smaller than the whale. And so, all of these kind of active behaviors are powered by the same machine. And so we are interested in sort of taking advantage of this hardware to program behaviors like cells achieve in vitro and also to build synthetic cells. And so you might ask, like, we know what this hardware is, what's the challenge? Like, why have we made more progress on information processing than we've made on mechanical control and mechanical regulation? And I would argue the challenge is kind of the fundamental way that these systems work almost evades our way to understand them. And so what are the problems? So systems like the spindle have many protein components. So eukaryotic cells have many, many different motor proteins. Important properties are motor proteins can actually act in opposition to sculpt structures like the spindle. There are lots of protein interactions. I think the really critical thing is this crossing of length scales. So you have nanometer-sized proteins generating structures that are 10,000, 100,000 times larger. The structures are very dynamic. Everything is consuming energy. And they're driven, because of that, way out of equilibrium. And so conventional theories of statistical mechanics that we might use to compute sort of the behavior of proteins or the self-assembly of structures near equilibrium don't really apply, and we don't really have good theoretical frameworks for studying systems like this that are consuming ATP and are sort of fundamentally dynamic in their behavior. And so over the last 10 years now, my research group has gotten interested in these reconstituted systems known as biological active matter as kind of a minimal way to get our hands on these mechanical machines of the cell and start using them for engineering applications. And so I want to tell you a bit about these active matter systems. And so as you guys all know well, there's a long history in biochemistry of reconstitution, right? So like much of what we know about things like the cytoskeleton, the cell cycle have come from reconstitutions in vitro of proteins, and we can use those reconstitutions to study the behavior of complicated processes like the cell cycle. The field of active matter biophysics has really pushed this to its limit. And so there's been a whole series of work, starting with work that has been inspiring to me from Stan Leibler and Francois Nedelec, where people have showed that you can take extremely simplified reconstitutions. Here, just two protein components, microtubules, which are polar structures. They're these filaments I told you about. They have minus ends and plus ends. And then Leibler and Nedelec took kinesin motor proteins. Each kinesin motor is itself actually a dimer. Each of those dimers walks along one microtubule filament, and they engineered these kinesin motor proteins very simply using biotin and streptavidin to link them together. So the critical thing Leibler and Nedelec did is they realized that the really critical way that cells get motor proteins to do self-organization and exert forces on microtubules is by linking them, because when these proteins are linked, they actually can walk on neighboring microtubules and pull them together. So they can exert sort of long-range mechanical forces in the cell. And what Leibler and Nedelec showed in this original active matter work about 20 years ago is that they could take just two proteins, microtubules stabilized, these kinesin motors that walk to the plus end of the microtubules, mix them with ATP, and get them to self-organize these structures which resemble microtubule asters in the cell that are used during cell division. But these structures are being self-organized in vitro, basically in water or buffer, with just these two protein components and energy. And so I just want to give you a little bit, we're going to talk a lot about these systems, so I want to give you a little bit of intuition what's happening. This is a simulation. And so the thing you could ask, like, why does this thing even form an aster? Why does this system do this? And the way you can think about it is you have motors walking along microtubules. Here's a simulation of this happening. The motor you don't see, but there's a link. And the green dots are the plus ends of the microtubules, and the motors walk. The plus ends get pulled together. So if you think about this happening in mass over thousands of microtubules, what's happening is the motors are all walking, all pulling. The motor proteins go to the center of the structure, the microtubules get pulled, the plus ends get pulled together, and you end up with this radially symmetric microtubule structure that we call an aster. So that's kind of the intuition. And so these active matter systems are really remarkable because you can get this to happen in a highly simplified setting, which is amenable to experiments and modeling. There's another thing that's really remarkable about these systems is that people have shown in ensuing years, so Zonover Dojic has shown that if you take the exact same system, microtubules, kinesin motors that are linked, ATP, and you add a crowding agent, polyethylene glycol, you drive the system into a dramatically different phase by forcing interactions between the microtubules. And so Dojic showed that PEG causes this microtubule system to generate this thing that they call an active nematic. What you see here are the microtubules, and they bundle and they beat in this incredibly kind of chaotic non-equilibrium fashion. And so the amazing thing about this active matter system is that you take something, you make one small change, and a totally new behavior happens. And so we have this kind of remarkable substrate for starting to do engineering. We have one system, it can do many different phases. The truth is that because of the things I told you about, the non-equilibrium nature, the complexity of crossing length scales, these phases have been sort of very poorly understood. So I'm just showing you right next to each other. Here's a movie originally taken by Stan Leibler and his group of their system forming these asters. Here's the same system beating. And the thing that we got interested in is sort of twofold. So one, this is an incredibly powerful resource for understanding the assembly and function of these self-organized microtubule structures. But there's been a problem with the system, which is that everything that happens sort of spontaneously. You mix the components together, you get these behaviors, but there's really has been no way to control it or interface it, either to understand how it's working or to re-engineer it. And so we started this kind of research that I'm going to tell you about with sort of a simple goal. We thought, okay, there's this paradigm in computer science, which is programming. We're interested in programming different systems. We want to program active matter. And if we are going to program, we need a way to read from the system. Well, we can do that through imaging, but we need a way to write to the system. We need a way to control the spatial and temporal activity of these motor proteins so that we can start to control what's happening, control self-organization, and ultimately to build programs. And so what I'm going to show you first is we took this system and we've brought it under optical control, and that's allowed us to build a set of active programs and sort of start to learn more about how these systems function and how they operate. And so what do we do? Okay, so our idea was really simple. So we have microtubules, we have this static protein link. What we decided to do is we were at UCSF, and a lot of people were playing around with these optically controlled dimerization systems. And so we decided to take this link, which is a static link, and just make it light switchable by trading out biotin and streptavidin for this protein system called ILID, which involves two components, ILID and SSPB. We attach these things to kinesin motor proteins, so we have motors, kinesin with each part of this. And then the key thing is that when we shine blue light on the system, ILID and SSPB link, and so we get a motor dimer, but we only get it in the presence of optical excitation. And so what this allowed us to do is we could take the Leibler system, but now we could shine a light, and we could get an aster to form at a specific location in time and space. And this is useful for a bunch of different simple reasons. So one thing is when you can start to control the system, it gives you sort of a much greater resolution to quantify its behavior. And so a historical issue with these active matter systems has been since everything's spontaneous, it's hard to sort of even quantify their dynamics over time. But now we have this kind of time-gated input. So we can turn on the light, form an aster, and then we can use image analysis to track the structure of the aster over time and study sort of different regions of the sort of different kinetic properties of the system. And so, for example, we've realized over time that the formation process is sort of actively driven by microtubule dynamics, but the disassembly process is diffusive. And we can show sort of in this movie that this process is also reversible. Okay, so there's basic quantification stuff you can do, but the more exciting thing for us was that when we coupled to the system, it allowed us to start sort of doing the thing that I'd said at the beginning. We could start designing programs. We could start getting the system to achieve different behaviors. And so we did this in a way that was sort of guided by simulation. Here's a simulation on the left-hand side. It's simulating the physics of the system, motor linking and pulling. And what we found is that we could shine and move the light, and we could get in simulation the aster to form and to chase this light. And then we can go back and forth between sort of simulation, not theory, and experiments. And we found that there's this sort of emergent property of the system, which is that following aster formation, the aster will actually chase a disk of light in a way that at this time was not entirely understood. So this is sort of an emergent property of these light-controlled active matter systems. So we've got this aster, this is a disk of light. And what we found is that as we pull the disk of light forward, the aster chases it, it lags the disk of light, and using this light-mediated motility, you can get an aster to move sort of hundreds of microns. And you can also do things like get the aster to move along curved paths. And so here you see an aster kind of dancing and fluctuating, and it's chasing this light disk around a spiral. And again, like you can see a lot of stuff in this movie, like the aster, there's new material coming in, but the aster is sort of maintaining some coherent structure. It's not like it's reforming at each point in space, but there are sort of microtubules that are being pulled along as part of this light-driven transport process. So here's what we call this combining programs. And so we form two asters at different locations, and then we can pull the light disk and cause the asters to move around the edges of a box. And so this was really exciting to us because this is exactly this kind of programmed manipulation that we'd been hoping to achieve, and we could achieve it using this sort of like very simple mechanism, which is just literally making this, being able to control the formation, the activity of these motors in time and space, sort of unlocked some of these emergent programs. And so I'll show you another one. This is what we call merge. And so this is simulation and this is experiment. And we found that if you form two microtubules and then you shine a light bar in the middle, the asters actually, they link and they contract and they pull together. And so that's a merge operation, and this is sort of reminiscent of a mitotic spindle. It's sort of an unstable mitotic spindle because a mitotic spindle is two asters with a stabilized middle. Here we have two asters that are unstable. And so we found that we could manipulate these microtubules, that we also found that as you manipulate the microtubules, they act on their surrounding environment and induce micron-scale fluid flows. And so people know that fluid flows are important in biology, for example, in microorganisms inside the cell during motility. And what we found as we started to manipulate these microtubules, I don't know if you can see it, you'll see it better on the next slide, is that in the background, when these microtubules linked, here it is with tracer beads. So now there are tracer beads in the system, and when the microtubules link, what you start seeing is the tracer beads start moving. And that happens because as the microtubules move, they act on the fluid in the environment and they drive fluid flows. And you can use these tracer beads to track the structure of these fluid flows. And so you can track the tracers using image analysis, and then you can change the light pattern. And what you see is that as you change the light pattern, you get sort of these very complicated flow fields with these vortices. And these flow fields depend on the geometry of the light pattern. And so you can give the system static patterns. Here's the dynamic pattern. We call this an active stir bar. And so what we're doing here is we're rotating the light bar, and you see a rotating flow field emerges. And this also got us interested and has gotten us interested in applications of this system for sort of technology, because one question is, there's a lot of microfluidic technology which uses like PDMS devices that are rigid, and this is starting to show that we might be able to use active matter to manipulate fluid flows on micron length scales in a very controlled way. Okay, so the way this research kind of played out is we had this very naive idea. Active matter is uncontrolled, we want to program it. Let's make it light switchable. Now that we have this interface, we were able to generate different optical programs. But the truth is, when we made these programs, we really had like no actual understanding of how these programs work. And so the interesting thing about the optical system is that you can now go in and start studying fundamental limits. And so, for example, one question we asked is how fast can you actually move an aster? And so you got to think about this aster structure. So it's a bunch of microtubules. It's highly dynamic. So there are motor proteins in the structure. The motor proteins are bouncing around. And what we found is that this red line is a little bit shifted. So there's an aster. Here's the light disk. we found that as you move the light disk faster and faster, the aster lags the light disk, and there's a gap that opens up. And you can sort of track this gap as a function of the speed at which you're moving the light disk. And the interesting thing is that this gap grows and grows and grows. And then there's a critical velocity, which in this system is about 400 nanometers per second. And at that critical velocity, the aster can no longer follow the light disk. So there's like a fundamental speed limit on aster motility. Now, at this point, the question is like, why does this speed limit exist? This is an emergent structure, self-organized structure. What in it, this speed limit is actually sort of near the motor velocity. The velocity of these motor proteins is about 1 micron per second. So you can ask like, maybe that's setting the speed limit of aster motion. But at this point, we had sort of very little understanding of how that speed limit is being set by the internal physics of the structure. I'll give you another one. So when you link the asters, you can track the velocity of the asters as they merge. And what we found is that as you make the initial distance between the asters bigger and bigger, they actually go faster and faster while they're linking, and you can accelerate the asters up to velocities of about 2.5 microns per second by making the linking distance go to a millimeter, which is actually like quite a large distance. And again, so there's this kind of velocity amplification as a function of linking. Another example of kind of a principle of an active program that at this point was poorly understood. And the same thing with the fluid flows. So we found this thing that we could change the geometry of the light excitation that would create a complicated microtubule structure. We can measure the fluid flows. The fluid flows in the system always have these complicated swirls and vortices. We can track the vortices, identify where they are, but had very little understanding of what sets the quantitative architecture of the flow fields and how the physics of this system is being transferred into the fluid. And so there's kind of this question. So we wanted to program active matter. We developed this optical interface. We developed these programs. And now there's this question which really gets us to understanding. So how are these programs actually working? What is the emergent physics of this microtubule system which is generating all of these behaviors and these speed limits? And so that's the thing we've been working on over the last couple of years. And so I want to talk with you about that. Okay, so the first thing we realized is that everything I've shown you so far is about these asters. We're forming these asters, we're moving these asters around. But what we realized is that all of the physics in this system and all the biophysics is actually being driven by a different structure, which you can barely even see here. And that is a microtubule network which forms in the background of the system upon light excitation. And you'll see it more here. So you'll see that we have these two asters. And now if you look inside the bar, the asters actually don't even interact directly. We shine light, and there's this network of microtubules that forms in the middle. And so what we found going forward is that all of the physics of these programs is actually being driven by this microtubule network that's forming in the background of the system, which is actually acting like the actuation device. And so using this light control, we developed an experimental system to actually study this kind of background microtubule network. Okay, and so what was the problem? So the problem with studying this network is if you take this system, so these are labeled microtubules that are motor proteins you don't see, we're shining a square light pattern on the system. And you see that initially what happens is there's this network that forms. But then the problem is that as the network contracts, there are these complicated fluid flows. And the network is sort of pulling in this flux of microtubule arms from the bulk in the background. And the problem for sort of understanding the physics of this system is you've got a lot of stuff going on. You've got microtubules contracting, you've got that dynamics, you've got this flow field. And it's hard to know like what the chicken or egg is, right? You've got the physics of the microtubule network, the physics of the flow, you've got transport. And so again, just to like speak to the power of what a lot of you guys are doing here, which is developing new engineering technology, we found out that we could use this light system to generate an optical illumination protocol which would sever the microtubule network from the background and really allow us to isolate its dynamics. And so what I'm showing you on the top is what happens for the constant illumination protocol. We form a network, the network contracts, we get these arms. we developed this dynamic illumination protocol. We form a network, but then what we do is we shrink the network, we shrink the light region with the network. And what happens when we do that is we sort of prevent the formation of these microtubule arms. And so then we can study sort of the pure physical behavior of this network. And I'm just going to show you this. I'm drawing these boxes, and so I'm actually not doing a good job of shrinking. You're just going to have to like bear with me. But what's actually happening here experimentally is we draw the box and then we shrink the box. And what you see is that when we do that, these arms do not form. And so you can just study the pure kind of mechanical dynamics of this microtubule network. And another kind of like fun aspect of it is we can change the geometry of this light excitation to make networks of different sort of sizes and shapes. You'll notice this interesting thing, which is like still has been puzzling us for a long time, which is as these networks contract, first of all, they contract, and that as they contract, they preserve their shape. And to this day, a question is this kind of shape preservation property like a special property of these non-equilibrium systems or something we could expect? I'm not going to talk about that, but I'm going to talk about sort of fundamental properties of this network. And so with this light protocol, what we could do is then use image analysis to track the density and the velocity of microtubules in this system as a function of time. So you can take the network, you can do image analysis to infer how material is floating, you can reconstruct this vector field, you can study the velocity, the magnitude, and direction of this vector field as a function of the position in the network that you're at so this is the position you're at relative to the center of the network. And we started to find sort of like very simplifying behaviors that provide kind of physical insight into how this network is working. And so the punchline for this network is the network actually is like a really simple object. So no matter what you do, you can make star-shaped networks, triangle networks, elliptical networks. It's a little bit hard to see these vector fields, but all of these networks just generate contraction. They just contract towards their center of mass. And they have maybe one important property, which is that as they contract, so you can make networks that are small, you can make networks that are big. No matter the shape, the velocity of the network is proportional to the distance of the point you're studying relative to the center of the network. So as you go away from the network just generates a linear velocity field whose magnitude scales linearly from the distance that you are studying from the center of mass. Okay, and so you study a 400 micron network, an 800 micron network, you look at the velocity of the contraction, they all fall on the same line. The 800 micron network is just contracting twice as fast at the boundary as the 400 micron network. And so you have this contractile network with a relatively simple property. There's a property that people study this is called telescoping. So what's happening in this system is that as this network contracts, it's pulling in. And so if you take one network and you add a second network, now if you think about contraction of this whole thing, this part of the network is being pulled. So if you look at one square, this boundary is being pulled by this much network. Now if I put two together, this boundary is now being pulled by twice as much network and sort of goes twice as fast. And then if you add another square, the network goes 3 times as fast. People call this telescoping, that as you add these network units together, they're all pulling on one another, and you get this sort of phenomenon of velocity amplification. And so this kind of like detailed study of the network biophysics yielded this like very simple insight, which is just that the behavior of these microtubule networks is contractile. That can be understood like just by extrapolating what I showed you for 2 microtubules to 100,000 microtubules. So for 2 microtubules, the plus ends, these green ends are getting pulled together. And so all of the points in the network are sort of getting squeezed and getting contracted. And this is what happens when you simulate the same dynamics over 100,000 microtubules, is that you just get all the plus ends getting pulled together. And the way you can think about this microtubule network is with a very simple analogy to a physical system that we're all sort of somewhat familiar with. And so you can think about the motor proteins as nodes in a mass spring network. And what's happening is when we light excite, these motor proteins are getting linked together and they're pulling. And so you can think about a microtubule network as a mass spring network that has some rest length, but that rest length is contracting as a function of time. And so you have springs, and all the springs are pulling together. That's happening at a constant velocity, which is set by the microtubule speed. And that leads to this just net contraction of the system internally towards its center. And this is a model you can make mathematical, so you can write down a model of masses and springs, you can simulate its behavior. This I'm just showing you that when you write down this model and you compare theory to experiment, you get very similar velocity fields, and you'll see it over here. This is velocity versus distance in theory and in experiment, and in both cases you get these really simple lines. And I'm just going to show you like one other thing. So just to talk about models, so the model I showed you on the last page is a model where we're studying like this mass spring network with these discrete links that are getting pulled together. So there's another way to study systems like this, which is from the perspective of continuum mechanics. And in continuum mechanics, what you do is instead of looking at particles, the microtubules and the motor proteins, you actually set up a bunch of fields. And so you study the velocity field of the network, the density field of the network. These kinds of continuum models, so this is del squared, so these equations are the governing equations of this network, which involve not particles, but sort of densities and velocities. And these equations have some nice properties because we were able to sort of define them and solve them. And again, like recapitulate this very simple behavior that the velocity of the network is predicted to scale linearly from the center of mass. And then we can use these models to do simulations like this. And we can also use these models to do simulations like this. And so these continuum models are nice because they extend to systems with more complicated geometries quite easily. And so what I'm showing you here is that we can use this continuum model to simulate the behavior of sort of a more complicated network geometry. Here in the background is the light pattern, and what you see for this network is it's actually predicted to sort of bend and deform as it contracts. And here's a comparison with experimental data so this is the simulation. You see this network bending and deforming. And now we can go to the experimental system, and we can compare theory to experiment, and we can sort of track, induce and track deformation patterns in these networks experimentally in a way that's predicted by these models. This whole business of like bending has been interesting to us because another thing that cells do is change their shape through internal deformations. And so we've been able to show that these networks can both contract but can also bend depend on their initial geometry. Okay, so how is this helpful? So now I just want to show you, okay, you've got this contractile network. How can this actually help us understand the behaviors that I showed you at the beginning of the talk? So aster motility. So this is how we think that asters are actually moving. And so we think that these asters there are sort of two important things happening at the same time. So when we light excite the system, we form an aster, and then we induce another light pattern. And we think what's happening is that second light pattern is actually forming a contractile network in front of the aster that pulls the aster along. And you can see this. So based on the model, we started doing higher resolution experiments where we image the motility phenomenon. And what you see is if you image at high resolution and frequently, you see that what happens is as the aster forms, there's almost this carpet in front of it as we move this light disk. And that carpet is a contractile network that is pulling this aster along. And the key thing that sets the speed limit I told you about at the beginning is that if you form this carpet on a time scale so that the aster has enough time to be pulled by the carpet, it can keep up and exhibit motility. But if you shine the light, it's a little bit hard to see, but if you move the light too quickly, this network doesn't have enough time to contract. And instead of generating motility, you generate sort of a contractile wave that just moves translationally through the system. And so we were able to develop kind of like even more simplified models of this phenomenon. You can think about this motility phenomenon in a really simple way. If you have a bunch of contractile particles that are all interacting, here's just a bunch of contractile particles, and now you turn on contraction in front of this structure, then what happens is you get a very similar carpet of contraction that pulls the structure along. And this is just a model we've abstracted out, the motor proteins extracted out the filaments that are just dealing with point particles that are attracting one another pairwise. And these models are useful because we can sort of like recapitulate the behavior. Here's transport. Here's the contractile wave regime. In this regime, there's actually no net transport of the structure. This light disk is just moving forward, generating contraction without transport. And the mental model you can think of is what's happening is the aster is kind of riding along in a potential well that's being generated by our light excitation. And so the aster's here, there's a potential well pulling it in, and then if this potential well moves too fast, the particle falls out and can no longer get pulled. And so kind of using that simple model, we've been able to compute an escape velocity for the aster. And one thing that's been challenging, that's always challenging in these systems is this crossing of length scales. So right now, we are not at the point where we can say, okay, the aster has this escape velocity, it's 400 nanometers per second, and that's set by the motor velocity. We have this escape velocity computed in sort of a more abstract way where we can say, okay, if you simulate this system, there are kind of two regimes of behavior. There's a regime in which you get motility, a regime in which you get contraction. As you make the stiffness of the attractions between the motor proteins stronger and stronger, the motility regime expands. Now what we're trying to do is sort of go down and actually connect this with the molecular details of the system and connect it with the molecular details of the motor proteins. But we've been able to do in these models is sort of generate a proof of principle for this kind of directed motility phenomenon and start to show that these different regimes can exist. So that's kind of like one example of using this contractile network to generate sort of mechanistic or sort of explanatory models. Another is we've been able to understand these fluid flows. So I'll just show you this. So here's a hexagon network. As that network contracts, it forms those arms I told you about. And what we can do using our contractile network model is we can compute the forces on the corners of this hexagon using the mathematical model. And then we can transfer these forces to the fluid. So when the microtubules act on the fluid, they cause fluid to move. And there's some equations which aren't really important called Stokes equations, which allow you to input a force and then compute the velocity of a fluid given that force. And so we can use those equations, and then we can compute a simulated fluid flow and compare that with the experimental fluid flow. And we've been able to see that the contractile network model is sufficient to allow us to determine the position of these different vortices that I told you about before. And so now, I don't know that we totally understand why they're occurring at different locations, but at least we can compute how the forces in the system generate vortices, and so we can predict how these vortices will move around as we change the geometry of the microtubule structure. Okay, so that's understanding. The last thing I want to show you guys is then engineering applications. And so the kind of logic of the whole thing is, want to understand these motor microtubule systems, build these programs. We developed this optical interface. Now we have the programs, led us to some fundamental physics, namely to understanding this motor microtubule network. And now what we want to do is use it to do some engineering, and I'll just show you a couple things that we're doing. So one thing that we're interested in is using these asters as kind of little agents that can move around and collect cargo. So you can imagine like micron scale transport's really hard. People want to do things like grab cells, grab beads, manipulate them. We have fax machines, but there are all kinds of limits of those fax machines. And so we've been exploring this paradigm of using asters as kind of a transport mechanism. And so I have a student, Enrique, and what he did is to do transport, there are all kinds of issues because of the physics of the contractile network. And so you have to pull, if you want to pick up beads with a light disk, you have to move the light disk, but you have to satisfy all kinds of constraints. You can't move it too fast, and you also can't, I'll show you here, have it backed intersect with itself. And so what we did is we built a little reinforcement learning agent, and what that agent does is it sits on the microscope, it images the system, and then it sends light patterns into the system. And the goal of the system is to do this kind of like Pac-Man game. So we train it on this simulation where what we're trying to do is train the RL agent to kind of use light to move around and collect a bunch of beats. And we've been able to implement this experimentally. So now you're going to see our little like active matter person. And the active matter person, so this is a bunch of microtubules, and that's the computer-controlled light. And what the light is doing is what the computer is doing is it's trying to move this light disk around to go and collect all the beads. And you see like it does an okay job, it makes some mistakes, like it misses this one. And practically we're actually interested in this as kind of a technology platform for doing sort of micron scale sorting of cells. More fundamentally, what this is doing is helping us understand sort of in even more detail the physics of the system, because you'll see like as it turns the corner, the structure of this changes, and there's actually an acceleration of the microtubule guy. And we think that has to do with some elastic properties of the structure as it bends. And so we're very interested in these kind of technology applications as a way to actually like push the system forward and also learn new physical insights. And then something else that I wanted to mention, because this like dovetails with some stuff that people are doing here, is what we really want to do is take this system and encapsulate it inside droplets. And we have this dream that we could make droplets and we could put transcription translation system inside these droplets. And then all we'd have to do is put DNA into the droplets and we could boot up the active matter system. So in the complete dream, we would have a droplet, we'd make motors and filaments, and then this droplet could be sort of like an autonomous device that could move around and do things. And we're only at the beginning of this dream. What we've been able to do is we've done a bunch of work over the last few years using TXTL, and we've been able to do some stuff where we like make different motor proteins in TXTL, and that's been really helpful for a bunch of other ways, a bunch of other reasons. And we have sort of like our first proof of principle result, which is going to seem very like simple and boring, but is like exciting to us, which is that this is a droplet of active matter, and it's sitting in oil actually. And then we can shine a light on it, and what happens is we observe that the droplet generates internal flows and spontaneously starts to move. And it actually moves pretty fast. So light excitation causes this droplet to start moving at about 10 microns per second, which is actually like not a bad speed given the speed that cells move. And so this is kind of like a very nascent thing, but I actually didn't think this would work because we had no reason to believe that the dynamics of active matter inside this droplet could actually couple to the physical environment and drive any kind of forward motion. Now what we're doing is we're going inside the droplet and we're trying to understand the flow field. So the question now is, clearly there's a flow happening inside the droplet. We think the flow field looks like this. One thing we don't understand yet is how that internal flow of material inside this droplet is generating the forward motion. It probably has to do something about the physics of the interface between the active matter and the oil environment. But I'm like super, super excited about this because like I really didn't think this would work. And I think now this kind of maybe justifies the fool's errand of trying to build this like synthetic cell thing. Okay. So the stuff I wanted to talk to you about, so there are like a couple of things I wanted to communicate today. The big picture things I wanted to communicate today is that I think this like motor filament active matter system, which has been studied a lot now in the kind of like biophysics domain, is actually an incredible resource for synthetic biology. And like, I want to convince you guys to go and think about mechanical and physical behaviors, even though they're like less NIH fundable these days, I can tell you. But I think are like super exciting aspects of life that we know very little about. And I also recommend, I was telling Scott, I found this essay from Kirshner, Mitchison, and Gerhardt from like 30 years ago called Molecular Vitalism, where they were trying to spur on the next generation of biochemists to sort of understand stuff like this in the face of genetics, which is like knocking out components. And so I think the motor filament system is like a great hardware for building mechanical programs, controlling mechanical programs. The main thing we've found is that by building programs, we've been led to sort of very detailed study of the biophysics of these motor microtubule networks. And these networks are actually simpler than we expected. They contract in ways that we can explain with modeling. And our future goal is really to get rid of the light and make this whole thing autonomous. The light is like a little bit of a parlor trick, and what we really want is to have droplets that can actually like sense and respond through synthetic gene circuits to their environment. And so with that, I'm just gonna mention that we have a really exciting team of people at Caltech that's working on this stuff. All this stuff is a very close collaboration between my group and the group of Rob Phillips. And there are a lot of people working on different aspects of the system, including like computational biology stuff and protein design. And we'd love, if people are interested, we'd love to have you visit because we really want to get people excited about this system. And I thank you very much for having me.
Questions? Oh, okay.
Hey, Matt, that was really cool. I have a question about your drops that could do forward motion when you shine light on them.
Yes.
If you do that to many, many droplets, do they all go in the same direction or do they just go in a direction? And how can you sort of control that to make them
Yeah,
autonomous?
that's a fantastic question. That like gets at the heart of the thing too, because you can't really see it here, but one thing is that it looks like there's a critical size. The droplets below a certain size don't move. The other thing is we think right now it is kind of like spontaneous symmetry breaking, where what happens is you shine a light and it goes in a direction. Now the question is like, can you control that by biasing the light pattern? We don't know yet, but I think there's reason. The good thing is that because this system is like a minimal system, you can predict the flow fields. So like you can compute these flow fields in 3D. So we're hopeful we can use that to sort of design light patterns to bias motion. It's a great question.
I had a question too. So I think you showed pretty spectacularly how just using preformed microtubules and one class of motor protein with the dimerization, you can generate a lot of complexity. I'm curious, two things that come to mind in terms of what might be missing, or not missing, but that cells have would be microtubules that are dynamically unstable,
Yes.
and how that might contribute to the behavior of these networks. And of course, the other is motors working in opposition, where
Yes.
the forces applied are either creating tension or compression, and how
Yes.
that might, do you think that would open up more complexity to the
Yes.
space?
So fantastic question. So like one thing that's been super frustrating about this system is you're like, it just kind of does this dinky contraction, right? You like do all this stuff and it's like, bloop, it just contracts. And so like one thing we really want to do is cells use opposing motors. And we think that cells use opposing motors in like a very interesting way. So what cells will do is like in the spindle, so this thing just contracts. And like one thing we really want to do is make things that can hold their shape, right? So forget contraction. Can you make a solid? Could you make a star? And we think that Scott's question like holds the secret, which is what the cell does is it jams the system by having one motor going one way, the other motor going the other way, kind of just like a kinase and a phosphatase, right? So you have like flux, and then you get a steady state. And so what the cell seems to do is it forms a network and it jams it, and that allows it to create a solid, but then switch the solid. And so one thing we really, really want to do, and would love to collaborate with anyone who's like remotely interested in this, is develop -n directed motors with light control or some other kind of synthetic control so that we could do exactly Scott's idea.
Thanks, Matt. That was a great talk. I actually had two questions. One was on the contractile networks you were showing.
Yes.
I was curious if there was anything about the negative space that you see
Yes.
that's actually what caught my eye. I was like, why is there like this negative space happening and nothing's happening to the microtubules surrounding? And that was one of my questions that I was really curious about, if you've dived into that.
Awesome question. And there's like another kind of thing just to say is the flows. Okay, so what we think is happening is that the network contracts, it's actually pushing out water, right? So like water, the microtubules are filling space, water we think is getting pushed out, and then the flow of the water kind of pushes out the bulk, these microtubules, and always generates this kind of circular depletion region. One thing that this has also made me think about, so we think it's the flow of water. One thing that's weird about this system, also relative to the cells, so like I applied for an NIH grant with some of this stuff and talked about flow fields, and people are like, flow fields don't matter in the cell. But when you do this stuff, you see flow happening everywhere. So you touch the microtubules and stuff flows. And so I kind of have this feeling like flow must be happening all the time, but maybe we don't appreciate it because it's really hard to image. And so like the answer to your direct question is it's fluid. And then generally I think like fluids are pretty interesting and cool mechanisms that might be interesting things to explore as well, even if they're challenging.
That was actually really cool. My second question was I'm thinking about your assay, the setup for your the microscope assay. Is there any heterogeneity in like the sample space itself? So am I assuming that you're just putting like a 1 microliter drop on a cover slide and putting the cover slip on top, and then is it just like a circle drop on the slide?
Yeah, it's like this. It's this glass sandwich. So it's like it's kind of a very primitive flow cell.
Yeah.
And there's a sandwich. So the spacing, it's like 70 microns between cover slips. And then we try to get sort of a nice two-dimensional slab of buffer, motor filament buffer.
So is there any heterogeneity in these contractions or the asteroids moving around where it's like near the edge of the drop versus the center of the drop? Like where you stimulate, if you stimulate in the center of the drop
Yes.
versus the edge,
Yeah,
have you
totally.
seen that heterogeneity?
Yeah, so there's heterogeneity. Where you are near the boundary like really can affect things and matter. The other thing that happens is, I don't have a slide for this, but there's a weird memory effect where like if you shine a light and form a network and then take the light away, there's more microtubule density in some region. And so then if you shine the light again, that region will form an aster and kind of remember where the aster used to be. And so this heterogeneity stuff is really, and like another thing about the heterogeneity is one thing that's weird is that there's like molecules underneath here and the molecules are fluctuating. And so you'll get, when the asters are moving, there are all these weird stochastic fluctuations that are being driven by kind of heterogeneous concentration densities, which are also really interesting, but we don't understand them very well.
So in developing the system, you say there's microtubules and kinesin. I'm curious how much you had to play with which microtubules and which kinesins
Yes.
and which PTMs and all of that
Yes.
that go, So,
So we would love to play. Okay, so that's been a long saga because this whole field was developed on one kinesin, K401, which Jeff Gellis at Brandeis truncated back from full-length kinesin to a minimal unit, and that's what everyone has worked with, and then cow tubulin. But we've gotten interested through this evolution question. So there's this weird evolution question of like no motors in prokaryotes, tubulin, sort of tubulin-like systems, but not tubulin in prokaryotes, and then all of a sudden in eukaryotes you have tons of motors, like people think the first eukaryote had 14 motors and microtubules. And so we started trying to bring more motors into the system. And we've tried 19 now, and we had trouble. And the reason was they're poorly characterized. And so we found that the motors have these interesting, and I was talking to Ivan about this, these inhibitory domains that fold over and block the motor domain. And so we're kind of working through bringing those in. One interesting thing is they can be very different. So there's a motor from an organism called Arcella that we brought into the system that has some like really kind of wild dynamics. Down here somewhere. Well, I'll tell you like another kind of wild thing is that when you even, so one thing we did, this, which I'll just show you really quickly because it's interesting, is that, okay, so this is our system. And then what we were doing is we were using this TXTL stuff, transcription translation, to make the motors. And so what happened is when we make our system in TXTL, instead of getting this, we get this. And so the cytoplasmic extract actually crowds the system and it generates an entirely new phase. And so we're trying to understand that phase. And then we found some motors that have actually really cool properties. And we're working on that. The thing that we have made no progress on is tubulin. And you can't make tubulin in E. coli. I think that, I mean, like we care about this, maybe a few other people care about this, but like for us, that would be a fundamental thing if we could make different types of tubulin and explore their behavior. So if you have ideas about that, that would be amazing. But it's been very limited.
I mean, it seems like you're at such the tip of the iceberg here.
Yeah.
I also, I saw your slide, that historical challenges. Why is mechanics so far behind, information? And informational processing has all those things. They have, I wrote them all down. Many protein components,
Yes.
many interactions. They're all the same.
Yeah, you're right. Far from equilibrium.
Over length,
Yeah.
also work over time.
Yes.
So I thought that slide was both.
Okay, that's good. So what do you think it is?
I think it's funding.
Okay, I'm with you. I'm with you. Okay.
That's what I think.
Okay.
I think that, with a lot of the information processing, there's a lot of disease relations and oncogenes and stuff, and there was a feeding frenzy that went in that. That's why.
Okay, I believe that. Yeah, it's hard to work on mechanics.
Maybe one short follow-up and then we'll call it. I just wonder if, Are there biochemical properties? So using the different motor isoforms
Yes.
is challenging for many of the reasons you said, right? That it's like maybe it's auto-inhibited, maybe it's just harder to purify or doesn't behave as well when folded. Like if you make point mutants or if you kind of more peck at like kinesin 401 and change ATP hydrolysis rates or things that affect kind of microscopic rate constants in the behavior of the motor, does that tune specific like the specific emergent properties of like or like what the thresholds are
Yes.
for certain things, and does that kind of give you a guide for how to bridge the nanoscale biochemical features of a motor to like what we might expect to fall out?
Yeah, I think that's a great idea. And there's like our friend Kurt Thorne, as a grad student, people have made point mutants of motors that change their speed. And there's a student who's put some of those in the system, and then you can change. This is like ongoing work, but aster size can be changed with motor speed and stuff. So I think that's a great, probably more controlled idea.
Right. Well, let's thank Matt for a very fun
Thanks, guys.
talk.